Today we will be starting with the ATP graph. I know many of your teachers or many schools skip this topic and you will regret it. So I decided to cover this topic. First of all, I also thought that to skip this topic, but when I saw the past papers, there was a frequent amount of questions on this topic. So we will be starting with the graph question. So this is the y-axis and the x-axis. First, there will be labelings. I have to label this graph, so give me any letters. I will take ABC. I will just take ABC. This is A. This is a enzyme activity graph. You all should know this is not a pH graph. This is not a temperature or the concentration graph. This is a enzyme activity graph. So you should all know this. So this is the A point where enzymes are present, but there are no substrate to form the process. Like you haven't eaten, your stomach is empty, but pepsin is there. The enzymes are there, but there are no substrate. There is no protein for the pepsin to digest. So this is the A point where there are only enzymes, no activity because there are no substrate, nothing to digest. So now you eat, you take a protein bar. For example, it went directly into your stomach. Stomach has pepsin, the activity increases. The activity increases, there are 20 enzymes. We will take the example of 20. There can be 100, there can be more than 100. Just take the example of 20 enzymes. There are 2 protein enzymes. 18 enzymes are still left. The activity increases. Then you eat another bite. The activity increases. Now how many enzymes are busy? Such as we can say 12 enzymes are busy. 12 enzymes are busy. You eat another bite, now a smaller bite, and there are all 20 enzymes, all are busy. There are no else enzymes. You eat another bite, it will stay in your stomach, but there is no enzyme to dissolve that substrate. So that substrate will wait for the first substrates to get digested, then it will go on that enzyme. That enzyme is not denatured, its active site is perfectly good, but why I'm saying it's sitting there? Because they are all the enzymes, such as there are 20 enzymes, and just take these are 20 enzymes, all have substrate protein on them, all are busy breaking down the protein. All are busy, all the 20 enzymes are busy. So now the speed will stop. The speed will stop, but the process will continue because that substrate is getting digested. The process hasn't stopped. It will continue. So it will continue now in a stationary pattern. What do I mean by stationary pattern? This is the point where the speed increases. Now there are no enzymes. All are busy breaking down the substrates, so the activity will go straight. So this point I will say this is B point. Just saying B point, it doesn't mean any else things. So now this, just we are thinking that all substrates are digested. We will say this point C. So the maximum speed that goes, this is the maximum speed. This is the maximum speed. Does it increases? Does it go anywhere? So this we will take this as the maximum speed. So to mention the maximum speed, we say V max. What I mean by V max, M-A-X, max velocity, maximum velocity. Maximum velocity. What do I mean by maximum velocity? The velocity won't increase. This is the maximum value. It's the 20. If you take a car example, the maximum velocity of a normal car, like a Pakistani car, will be like goes to 200 miles per hour. So that's the maximum speed it can go. So this was the maximum speed these 20 enzymes can go. All the enzymes get busy at this point. So what we say, this is the saturation point. What do I mean by saturation point? When all the enzymes are busy, there is no enzyme left. All this is, we say, this position we say saturation point, when the speed is not increasing, but the process is continuing. The process is continuing, but the speed isn't increasing because all the enzymes are busy breaking down the substrates. So this is known as the saturation point. I hope you all understand. This was just the basics. We all have to know this. If you want me to more label it, I would say this area is known as available. We would say this area as available. Why I'm saying this process available? Because the enzymes are available, but the proteins, but the substrates are less at this point. Why they are less? You eat first bite, there were two proteins in it, so the enzymes, the activity increases, but the enzymes are still there. Then you eat a bite that has 12 enzymes, the activity greatest increase, it has a great increase. The enzymes are still there. How many enzymes? 6 enzymes. Then you take a bite, 6 proteins were in there. So at this point when you eat the 12 proteins bite, so the enzymes were available. So how can we explain this graph? The graph begins at the A, the enzymes were available, but as the diet contains the protein, as the diet enters, the enzymes were available, and the speed increases and the enzyme activity increases. As it reaches its maximum velocity, the graph becomes stationary. This point is known as saturation point, because there are many substrates but less enzymes, because all enzymes are busy digesting the substrates. I hope you all understand this basic concept. You all should focus on this graph. Alright, thank you, bye.